Hello student, good afternoon. Today we will start the glycolysis pathway. Glycolysis pathway. Glycolysis pathway. The 10 reactions of the glycolysis pathway are given in figure. Follows this diagram as you read about the individual reactions in the figure. Glucose and fructose are drawn in their linear forms so that you can see more easily the reactions that occurs at each step. The first reaction is the phosphorylation of glucose by ATP catalyzed by hexokinase. ATP catalyzed by hexokinase. This enzyme is found in all cells and has a high affinity low Km for glucose. Its affinity for other hexoses is significantly lower because the phosphate comes from ATP. This reaction is irreversible under cellular conditions. The liver contains an additional enzymes, glucokinase, which has a high Km for glucose. This enzyme is capable of phosphorylating glucose only under conditions of high glucose concentration, such as after a large meal. Phosphorylation of glucose is important both as the first reaction in the glycolysis pathway and for preventing glucose from being transported out of the cell. Very little free glucose is found inside the cell because of the high activity of the phosphorylating enzymes and the irreversibility of the reactions. Because the transport system for glucose has a low affinity for glucose 6 phosphate. Phosphorylation prevents glucose release from cells by converting glucose to a form that is not readily transported. The next step in the pathway is the isomerization of glucose 6 phosphate to fructose 6 phosphate. This is a readily reversible reaction that changes an aldopyranose glucose to a ketopyranose fructose. The enzyme catalyzing this reaction is phosphoglucose isomerase. The third step is the phosphorylation of the fructose 6 phosphatase to fructose 1,6 diphosphatase by phosphofructokinase. This reaction uses ATP as the source of the phosphate. Phosphofructokinase is an important regulatory enzyme. It catalyzes the first committed step towards the oxidation of glucose by the glycolysis pathway. In the next step, for fructose 1,6 diphosphate is cleaved by aldolase to 2-triose phosphatase, glyceraldehyde 3-phosphatase and dihydroxyacetone phosphate. The 2 trioses are interconvertible in a reaction catalyzed by triocephosphate phosphate, triophosphate isomerase. The equilibrium of the reaction favors the formation of dihydroxyacetone phosphate because glyceraldehyde 3 phosphate is continuously being removed by the next step in the pathway, reaction 6. The reaction proceeds in cells in the direction in which it is written in figure. The next reaction 6 is the oxidation and phosphorylation. Glyceraldehyde 3-phosphatase by NAD to produce 1,3-diphosphate phosphoglyceride. Glyceraldehyde 3-phosphate dehydrogenase catalyzes this reaction with uses inorganic phosphatases, not ATP, as the source of phosphate. The produce is a mixed anhydride that has a large negative Dg for hydrolysis. Therefore, 1,3-diphosphoglycerate is a high energy compound. This is your first encounter with a reaction that uses nicotinamide adenine dinucleotide or NAD. Let us digress a moment to discuss the important compound. NAD is made up of the base nicotine, amide, ribose and ADP. A second phosphate may be attached to the hydroxy group at the carbon, creating NADP. NAD and NADP serve as a mobile electron acceptors for a number of oxidation reduction reactions. They usually are not covalently bound to the enzymes with which they react. They reduce forms NADH and NADPH. Dihydronicotinamide adenine dinucleotide phosphate have the structure shown in figure. In the reaction 7-phosphoglycerate kinase transference the high energy phosphatase from 1,3-diphosphoglycerate to ADP, producing the 3-phosphoglycerate and ATP. This reaction is a substrate level phosphorylation. It is an unusual reaction in metabolism because most reaction producing ATP utilize the electron transport chain in the mitochondria rather than the transfer of phosphate from high energy compound that is not nucleotide. Enolase catalyze the dehydration of 2-phosphoglycerate 9,2-phosphonopyruvate and another high energy compound phosphoenolpyruvate, then transfers phosphate to ADP in reaction 10 producing pyruvate and ATP. The enzymes catalyze this reaction is pyruvic kinase, which is the named for the reverse reaction. NADH produced by the reaction normally is oxidized back to NAD in the mitochondria. If this does not occur, NAD can be regenerated in the cytoplasm from NADH by reducing pyruvate to lactate in a reaction catalyzed by lactate dehydrogenase LDH. By utilizing this reaction, glycolysis can continue even in the absence of oxygen. In the presence of oxygen, pyruvate is decarboxylated and the 2-carbon fragment is oxidized further in the mitochondria to carbon dioxide and water. This is the pathway of glycolysis pathway. Okay, thank you.